Cancer is a complex disease that is characterized by distinct cellular and molecular changes. Understanding the key hallmarks, or defining features, of cancer is critical for developing effective treatments and diagnostic tools. This slide provides an overview of the hallmarks of cancer, which will be explored in more detail in the following slides. The image on the right provides a visual representation of the molecular biology of cancer. I'll use this as a reference point to highlight the key hallmarks as we go through the presentation. The key hallmarks of cancer we'll be discussing today are the abilities of cancer cells to sustain proliferative signaling, evade growth suppressors, resist cell death, and enable replicative immortality. Sustained proliferative signaling refers to how cancer cells acquire the capacity for continuous growth and division by deregulating normal growth signaling pathways. Evading growth suppressors involves cancer cells bypassing the mechanisms that normally restrict cell growth and division. Resisting cell death means cancer cells are able to resist programmed cell death, or apoptosis, and continue growing uncontrollably. Enabling replicative immortality is the ability of cancer cells to maintain telomere length, giving them the capacity for limitless replication. These hallmarks represent the fundamental biological capabilities acquired during the development of human cancers. Cancer cells have genetic mutations that disrupt normal growth signal regulation, leading to uncontrolled cell division and rapid proliferation. Cancer cells can stimulate their own growth by secreting growth factors or hijacking the signaling of surrounding cells in the tumor microenvironment. Overexpression or constitutive activation of growth factor receptors on the cell surface can drive sustained proliferative signaling in cancer. Dysregulation of intracellular signaling pathways, such as the PI3K/ACT and RAS/MAPK pathways, can amplify and prolong proliferative signals in cancer cells. Cancer cells can disrupt the normal signaling pathways that regulate cell growth and division, allowing them to proliferate uncontrollably. Cancer cells can bypass the checkpoints and controls that normally regulate the cell cycle, enabling them to divide and replicate without restraint. Tumor suppressor genes, which normally act as brakes on cell growth, become mutated or silenced in cancer, allowing unchecked proliferation. This slide illustrates how cancer cells evade the normal growth-regulating mechanisms, leading to uncontrolled cell division and tumor formation. Cancer cells have developed ways to resist programmed cell death, or apoptosis, allowing them to survive and grow unchecked. They often increase the expression of proteins that block the apoptotic signaling cascade, tipping the balance towards cell survival. Mutations or inactivation of the p53 tumor suppressor gene, a key regulator of apoptosis, is a common feature in many cancers, further enabling their resistance to cell death. Cancer cells are able to avoid the normal limits on cell division by maintaining their telomeres, the protective caps at the ends of chromosomes. This is done through the activation of telomerase or alternative lengthening mechanisms. Healthy cells typically can only divide a limited number of times before entering a state of permanent growth arrest called senescence. Cancer cells have acquired the ability to bypass this natural barrier to immortality. Tumors stimulate the growth of new blood vessels, a process called angiogenesis, to supply oxygen and nutrients needed for rapid tumor growth and expansion. Tumors secrete vascular endothelial growth factor, VEGF, and other factors that bind to receptors on endothelial cells, triggering them to proliferate and migrate to form new blood vessels. The new blood vessels sprout, branch, and remodel to create a dense network that supplies the growing tumor with essential oxygen and nutrients, fueling its progression. This slide provides an overview of the key mechanisms underlying tumor angiogenesis, which is a critical process that enables solid tumors to grow and metastasize. Cancer cells undergo a transformation from an epithelial to a mesenchymal phenotype, which enhances their migratory and invasive capabilities. Cancer cells secrete proteolytic enzymes that enable them to breach the basement membrane, a barrier that normally confines cells. Reorganization of the cancer cell cytoskeleton and acquisition of a more migratory phenotype allow them to actively invade surrounding tissues. Cancer cells can enter the bloodstream, intravasation, and exit at distant sites, extravasation, enabling metastatic spread to other organs. Cancer cells often exhibit widespread genetic changes, including chromosomal instability, aneuploidy, and accumulation of DNA mutations. These genomic abnormalities drive the hallmarks of cancer and allow cancer cells to rapidly evolve and acquire new capabilities. Defects in DNA repair pathways, exposure to carcinogens, and errors during DNA replication can lead to the acquisition of driver mutations. These driver mutations confer a growth advantage to cancer cells, allowing them to outcompete less fit cells. As cancer cells accumulate genetic changes, they undergo clonal evolution, with more aggressive, drug-resistant subclones emerging. Genomic instability is a key characteristic of cancer cells that promotes tumor progression and metastasis. Chronic inflammation can create a microenvironment that supports tumor growth, angiogenesis, and metastasis. Immune cells like macrophages, neutrophils, and lymphocytes can be co-opted to promote cancer progression. Cytokines, chemokines, and other inflammatory signals can stimulate proliferation, survival, and migration of cancer cells. Inflammation can also induce genetic instability and suppress anti-tumor immune responses in the tumor microenvironment. This slide provides an overview of how tumor-promoting inflammation can drive various aspects of cancer progression. Cancer cells exhibit a higher rate of glucose uptake and glycolysis, even in the presence of oxygen. This is known as the Weinberg effect. Many cancer cells have impaired mitochondrial function, leading to a shift towards glycolysis as the primary energy-producing pathway. The excess pyruvate generated from increased glycolysis is converted to lactate, which is then secreted by cancer cells, creating an acidic tumor microenvironment. Cancer cells can exploit immune checkpoint proteins like PD-L1 to suppress T-cell activity and avoid detection by the immune system. Tumors release immunosuppressive factors that inhibit the function of cytotoxic T cells and natural killer cells, hindering the body's ability to mount an effective anti-cancer immune response. The tumor microenvironment is often characterized by an abundance of regulatory T cells, myeloid-derived suppressor cells, and other immunosuppressive cell types that create a hostile environment for anti-tumor immunity. Genetic changes in cancer cells can lead to the downregulation of major histocompatibility complex (MHC) molecules, preventing the proper presentation of tumor antigens to the immune system. Cancer cells often have genetic mutations that disrupt normal cellular processes, leading to uncontrolled growth and survival. Many cancer cells exhibit chromosomal abnormalities, such as aneuploidy and chromosomal rearrangements, contributing to genetic heterogeneity. Alterations in DNA methylation, histone modifications, and chromatin remodeling can dysregulate gene expression and drive cancer progression. These genetic and epigenetic changes collectively contribute to the development and progression of cancer. Oncogenes are genes that, when mutated or overexpressed, can drive uncontrolled cell growth and division, leading to cancer. They hijack the normal cell signaling pathways that regulate proliferation. Tumor suppressor genes normally act as brakes on the cell cycle, preventing uncontrolled growth. When these genes are mutated or lost, cells can divide without restraint, leading to tumor formation. The balance between oncogenes and tumor suppressors is crucial for maintaining normal cell growth and division. Disruption of this balance is a hallmark of cancer development. Cancer cells often have abnormal chromosomes, like extra or missing chromosomes, as well as rearrangements in their chromosomes. This chromosomal instability is a hallmark of cancer. Many cancer types also exhibit microsatellite instability, where small, repetitive DNA sequences accumulate errors due to defects in the DNA mismatch repair system. Impaired DNA damage repair mechanisms, such as nucleotide excision repair, contribute to the genetic instability seen in cancer cells, allowing more mutations to accumulate. Changes in telomere length and telomerase activity also drive genome instability in cancer, allowing cancer cells to replicate indefinitely. These various forms of genetic instability are key features that distinguish cancer cells from normal, healthy cells and contribute to the rapid evolution and progression of the disease. Cancer cells often exhibit abnormal chromosome numbers and structures, leading to genomic instability. Aneuploidy, the presence of an abnormal number of chromosomes in a cell, is a hallmark of many cancer types. Defects in cell division processes, such as mitotic dysfunction, can cause unequal chromosome segregation and lead to aneuploidy. The image on the slide shows a microscopic view of a cancer cell undergoing mitosis, with abnormal chromosome segregation and an uneven distribution of chromosomes, illustrating the concept of chromosomal instability and aneuploidy in cancer. Cancer cells exhibit abnormal DNA methylation patterns, leading to silencing of tumor suppressor genes and activation of oncogenes. Altered histone acetylation and methylation disrupt the normal gene expression programs in cancer cells. Changes in chromatin structure and accessibility affect gene regulation and genome stability in cancer cells. Aberrant expression of microRNAs and long non-coding RNAs can dysregulate key cellular pathways in cancer. The tumor microenvironment is a complex ecosystem made up of various cell types, including cancer cells, immune cells, blood vessels, and supporting stromal cells. These different cell populations interact through complex signaling networks to influence key processes like angiogenesis, immune evasion, and metastasis. The extracellular matrix surrounding the tumor also plays an important role, providing structural support and modulating tumor cell behavior through biochemical and biomechanical cues. Understanding the intricate composition and signaling dynamics of the tumor microenvironment is crucial for developing effective cancer therapies that can target multiple aspects of this complex system. Tumors stimulate the growth of new blood vessels, known as angiogenesis, to supply oxygen and nutrients. These tumor blood vessels are often irregular, leaky, and inefficient compared to normal vasculature. The tumor vasculature is influenced by a range of stromal cells, including immune cells, fibroblasts, and parasites. These stromal cells can either promote or inhibit the growth of the abnormal blood vessels. Tumor blood vessels are constantly undergoing remodeling, with new vessels forming and existing ones regressing. This vascular remodeling happens in response to the changing needs of the growing tumor. The immune system tries to fight cancer by sending T cells and other lymphocytes to infiltrate the tumor, but the tumor often finds ways to suppress or evade this anti-tumor immune response. Myeloid-derived suppressor cells accumulate in the tumor and inhibit the function of T cells and other immune cells, allowing the cancer to evade detection. Tumor-associated macrophages recruited to the tumor site can be educated by the tumor to promote cancer progression, angiogenesis, and metastasis instead of attacking the malignant cells. Regulatory T cells accumulate in the tumor and suppress the anti-tumor immune response, further enabling the cancer to grow unchecked. The tumor microenvironment is a complex ecosystem where stromal cells play a critical supporting role for cancer cells. Stromal cells provide structural and functional support, allowing cancer cells to grow and progress. These cells communicate with cancer cells through intricate signaling pathways, influencing their behavior and survival. Stromal cells secrete and remodel the extracellular matrix, creating a permissive environment for tumor growth and metastasis. Stromal cells, such as cancer-associated fibroblasts, also promote the formation of new blood vessels to supply the tumor with nutrients and oxygen. Understanding the role of stromal cells in the tumor microenvironment is crucial for developing effective cancer therapies that target these supportive cells. Cancer cells undergo dramatic metabolic changes to fuel their rapid growth and survival. A key feature is the Weinberg effect. Cancer cells preferentially use aerobic glycolysis for energy production, even in the presence of oxygen. This leads to high levels of lactate production, which is a hallmark of cancer metabolism. Cancer cells also become addicted to glutamine, relying heavily on it for biosynthesis and other critical metabolic processes. These metabolic reprogramming events provide cancer cells with the energy and building blocks they need to proliferate uncontrollably. Cancer cells exhibit an increased reliance on glucose as their primary energy source, even when oxygen is available. This preference for glucose over more efficient mitochondrial respiration is known as the Weinberg effect. Cancer cells convert glucose to lactate through a process called aerobic glycolysis, which is less efficient for energy production but may provide other benefits. The Weinberg effect involves the suppression of mitochondrial respiration, shifting the cell's metabolism towards this aerobic glycolysis pathway. This metabolic reprogramming is a hallmark of many cancer cells and is an area of active research in understanding cancer cell biology. Cancer stem cells have the remarkable ability to self-renew, allowing them to maintain and propagate the tumor over time. These cells can differentiate into diverse cell types within the tumor, contributing to the heterogeneity we see in many cancers. Cancer stem cells are believed to be the root cause of tumor formation and growth, making them a critical target for cancer treatment. Sadly, these cells are often resistant to conventional cancer therapies, leading to relapse and metastasis in many patients. Understanding the unique properties of cancer stem cells is crucial for developing more effective and lasting treatments. Epithelial mesenchymal transition, EMT, is a crucial process where epithelial cells transform into a more migratory and invasive mesenchymal phenotype. During EMT, epithelial cells lose their cell-cell adhesion and polarity, down-regulating important structural proteins like E-cadherin. As epithelial cells undergo this transition, they acquire mesenchymal characteristics, expressing markers like vimentin, N-cadherin, and fibronectin. These mesenchymal traits enable increased motility, invasion, and resistance to apoptosis in the transformed cells. The image on the slide illustrates this epithelial to mesenchymal transition, showing cells in various stages of transformation. Cancer metastasis is a complex process involving multiple steps. First, cancer cells break through the basement membrane and invade the surrounding tissue. This is known as local invasion. The cells then enter the bloodstream or lymphatic system, a process called intravasation. Next, the cells exit the circulatory system at a distant site, which is called extravasation. Finally, the cells proliferate and form new tumors at this secondary location. This is metastatic colonization. Understanding this stepwise process is key to developing effective treatments to prevent the spread of cancer. Solid tumors require a dedicated blood supply to obtain oxygen and nutrients for growth and survival. Tumor cells secrete proangiogenic factors that stimulate the formation of new blood vessels, a process known as angiogenesis. The blood vessels that form in the tumor microenvironment are often abnormal, with irregular branching, leaky walls, and inefficient blood flow. This chaotic and hypoxic tumor vasculature contributes to the challenging nature of the tumor environment. Cancer cells have developed various mechanisms to evade detection and destruction by the immune system. One way they do this is by masking their antigenic markers, making it harder for the immune system to recognize them. Tumors can also release immunosuppressive factors that impair the function and proliferation of immune cells, dampening the immune response. Cancer cells can activate immune checkpoint pathways, like PD-1/PD-L1, which inhibit T-cell responses and allow the tumor to grow unchecked. Genetic instability in cancer cells can lead to the loss or alteration of immunogenic antigens, further hindering immune recognition. The key focus of this slide is the therapeutic targeting of the hallmarks of cancer. It highlights three important approaches. Personalized therapies that target the specific hallmarks present in an individual patient's cancer. Combination approaches that target multiple hallmarks to overcome cancer's redundancy and adaptability. Emerging innovations like immunotherapies, nanomedicine, and gene editing that open up new ways to target the hallmarks. These approaches can lead to more effective and tailored cancer treatments. This slide is visually striking, with a laboratory scientist examining a molecular model and glowing DNA strands in the background, conveying the scientific and technological nature of these advances. Precision medicine is a key focus area, developing personalized treatments tailored to individual genetic profiles and tumor characteristics. Immunotherapy breakthroughs are enabling us to harness the immune system more effectively to target and eliminate cancer cells. Combinatorial therapies, where we explore synergistic effects by combining multiple targeted therapies and immunotherapies, hold great promise. Early detection innovations, such as advancing diagnostic tools and biomarkers, are critical for earlier cancer detection and screening. The hallmarks of cancer have enabled researchers to better understand the complex mechanisms underlying tumor development and progression. Continued exploration of these hallmarks will inform future advancements in cancer detection, prevention, and treatment. Addressing the hallmarks of cancer requires a multifaceted, collaborative approach involving researchers, clinicians, and other stakeholders. By working together, the scientific community can drive more effective and comprehensive cancer management strategies. Understanding the unique hallmarks of an individual's cancer can inform the development of more targeted and personalized treatment approaches, leading to improved patient outcomes and quality of life.